Hi, it's Miss Parrott, and this video is the first in the series AP Bio Exam Skills. In this series, we're going to be looking at different types of classic AP Bio questions. We're going to be looking at the same idea presented in several different ways and focus on how to interpret and answer AP Bio multiple choice and FRQ questions. So, here is our classic cell in a beaker. We're going to be looking at water potential, osmosis, tonicity. I have videos in my cell biology playlist and in the AP Bio equations explain list that cover all kinds of stuff about these topics. This one we're going to focus on how to think through questions and how to answer questions. So feel free to check those out for more information. But a quick review of the basics. One is that water is always going to move from an area of high water potential to low water potential. Solute potential is always negative, and pressure potential is 0 in an open container for animal cells, not necessarily for plant cells because they have a cell wall which will give them some pressure potential. And another quick review of tonicity. So, remember, all solutions are made of solutes and solvents. The major solutes you're going to see in AP Bio are salt and sugar. That could be sucrose, glucose, any kind of oats. And then the solvent, since we're looking at living systems, is most often water. You're going to see comparison words like hypertonic, hypotonic, and isotonic. Again, I have a whole video on tonicity that you can click, find, and practice more with that. But basically, a hypertonic solution is a solution that has a higher solute concentration compared to another one. So in this picture, you see a cell in a beaker in a hypertonic solution because there is a higher solute concentration outside the cell than inside. This one would be considered hypotonic because it has a lower solute concentration, but therefore higher water concentration than inside the cell. So water is going to move from high water to low water concentration. So it's going to move in. In an isotonic solution, the solute concentrations are equal inside and outside of the cell. But that doesn't mean water doesn't move at all. It just means, and you may see these words, no net movement of water. It's a dynamic equilibrium because the molecules are always moving. There are 4 main ways that you're going to see solutions described in the AP Bio exam. It could be by tonicity, hypertonic, hypotonic, isotonic. Molarity, so you might have a 0.2 molar sucrose solution. Molar, this capital M, you may also see moles per liter. It means the same. It's just the unit broken down into its pieces. You may see it written as a percent, like a 5% salt solution, or you may see the water potential actually described. You'll know that it's describing water potential if it uses the units of bars or megapascals, like negative 0.3 bars or negative 0.5 megapascals. So since there are all these different ways to describe it, and some of them describe the concentration of water, like water potential, more than like molarity that describe the solute concentration, it can get a little confusing when we're reading questions. So first, let's distinguish what makes an area high water potential versus low. So an area with high water potential is going to be less negative than a lower one. It's going to have a lower solute concentration and a higher relative water concentration compared to the other solution. Whereas an area with low water concentration will be more negative. That can get tricky sometimes. But it will have also more solute. So how does this look like when we break it down into our 4 ways of describing? So if we're looking at water potential as bars or megapascals, an area of high water potential will be less negative. It may still be negative, lower than 0, but it'll be less negative, closer to 0 than the other. So in this case, negative 0.2 bars is higher than negative 0.4 bars. So don't get tricked up. Just because the number 4 is bigger than the number 2, negative 0.2 is closer to 0. So this is the higher area, the higher water potential area. So water will move from here to here, high to low. Also with water potential, pure water with no solute and no pressure is 0. So you could have water moving from an area of pure water to an area with some solutes. So 0 to even if it's a small number like negative 0.1, if it's moving from 0, it's less negative, or it's not negative at all, to the negative area. That's less. If we look at molarity and percent concentration, these both look at the amount of solute. Okay, so an area with higher water potential, you can also kind of, not exactly the same, but in a ballpark same, would be like high water concentration. Okay, so a 0.5 molar solution is then going to have a higher water concentration than a 0.8 molar. 0.8 molar is going to have more solute in it, so it's going to have a lower solvent or water concentration. So water will move from here to here. Again, even though 0.8 is higher than 0.5, this is talking about solute, not water. And water is what's going to move from high water potential to low water potential. The same is true with a percent solution. A 5% sugar solution is more sugary and therefore less watery than a 3% sugar solution. It has less solute and a higher water concentration. So water will move from an area with a 3% solution from an area where it's 3% solute to an area where it's higher. Higher solute, not higher water, lower water. And finally, tonicity. So I have a whole video on tonicity. Check it out. Hypotonic. So I like to think of like hypo is like a hippo, and hippos, you always see them like with their heads under barely sticking out, most of their body submerged in water. So hypo, hippo, lots of water around, and they're kind of swollen up, right? They're like these big round guys. So water will move from an area that's hypotonic, higher water concentration. Hypotonic means low solute concentration, lower, to a hypertonic area. So you got to pay attention to how it's worded, if the wording focuses on the water concentration or the solute concentration. And if so, what does it tell you without telling you about the other piece of the puzzle so that you can figure it out? Here's a strategy for interpreting and answering the following questions. We're going to look at 3 examples, just a simple one, and then a multiple choice, and then an FRQ. So stick with me or fast forward ahead to the kind of question you want to know about. But the strategy is, step 1, take the words and turn it into a picture. Or if it's picture, turn it into words or label it. Add that information that's there but maybe not presented obviously to you. We're going to do this by first drawing it, our classic cell in a beaker. We're going to add dots for solutes. And if it's a plant cell, we're going to add a cell wall. That's going to matter because it's going to impact how the shape changes at the end. Then we need to consider, based on what we just looked at, where will water move? We know it's going to move from high to low water potential, but where's the high area and where's the low area? We got to figure that out. Then let's draw an arrow, like physically draw on your picture to be sure that that makes sense. And then to answer your question, add whatever other super sassy science words need to be added to make it make sense. All right, let's look at question 1. This is an easy one. So we're going to look at it in both animal and plant cells. So if we have a beaker with a cell, animal cell with a 0.1 molar cytoplasm and a 0.2 molar solution in the beaker, what's going to happen? Which way will water move? So, right, draw it by, we did this part, we labeled. You can add dots. Okay, so 0.2 molar outside and 0.1. So really we just need to put more dots outside to show a higher solute concentration than inside. Okay, so the inside of this has much more relative open space than the outside does. So water is going to move from a higher water concentration, that's this, so 0.1 molar, remember, that means it has less solute. 0.1 is less solute, so more water concentration inside than outside. So we're going to draw our arrow, and does that make sense? Yes, because remember, the goal of water movement is for the solutions to become isotonic, or to reach a dynamic equilibrium. So if water leaves the cell, is that going to make the outside more watery and more like the inside? Yeah. So it's trying to make it equal. So our arrows there, it makes sense. Which way will water move? Water will move from, let's add some super sassy science words, the hypotonic inside of the cell into the hypertonic solution outside of the cell, causing the cell to shrivel or to shrink. In a plant cell, if we have the same situation, our water is still going to move out, but the cell wall is not going to collapse. It's made of like a rough cellulose structure, but it's also going to shrivel. But there is a super sassy science word for the cell membrane of a plant cell shriveling and pulling away from the cell wall, which would result in wilting. That's called plasmolysis. Plasmolysis, when the cell membrane pulls away from the cell wall. So be sure you recognize your super sassy science words, because otherwise they may throw you off and make you second guess yourself. Next question, multiple choice. This one gives us a data table, and then it says what's in the data table in writing. A plant cell has a total water potential psi of negative 0.8 megapascals. It is placed in a solution with water potential of negative 0.4 megapascals. What happens to the cell? So, step 1, turn words into a picture. So I went ahead and set up the drawing. So, and what's great, this one already gives us water potential, so we don't have to be thinking too much. But we need to label. So our plant cell is negative 0.8. And then our beaker is negative 0.4. Now we don't need to draw the dots because, I mean, you can if you want. It would be more on the inside, right? The more negative it is, usually the more solutes it's got going on for it. But in this case, we can just see. The outside is the area of high water potential. It's closer to 0. It's less negative. And the inside is the area of low water potential. So we're going to draw the arrow. Yep, that makes sense. Water is going to go in to try and even it out. And so, which of these shows water going in? We have water moves into the cell and turgor pressure increases. Possibly. B, water moves out of the cell, becoming flaccid or floppy. Nah, that's not it. C, water moves out and the cell lyses. That means the cell explodes. No. Partially because we have a plant cell, and no, water is not moving out. And then D, there's no net movement. That would be the case if it was an isotonic solution. But we have a difference, so there is going to be movement. That won't be it. So A, yes, water will move into the cell, and turgor pressure, or the pressure of the fluid, the cytoplasm on, let's make it a plant cell, or the cell wall, that pressure is going to increase because there's more water pushing. All right, now for the FRQ. All right, a lot of times FRQs give an example of experiment. They tell you about the results, and then they ask you to explain why it's happening and maybe predict something or do a calculation. We're not going to calculate in this one. I've got a whole calculation situation in another video. Let's look at this. A student places plant cells with 2% NaCl cytoplasm. So that means inside the cell, there's a 2% salt solution. And it places those cells into solutions of 0%, 2%, and 5% salt. So we have 3 beakers. All right, we have 3 cells. They are all 2% inside. 2%, 2%. This one has no solutes. It's a 0% salt solution. 0%. This one has a 2%. And then this one has a 5%. Okay, cool. Over time, cells in the 0% become turgid, right? So again, they're plant cells, so they're going to swell up, they're going to fill up. So that means water went into them. So I could draw an arrow, water filled up, and it's not going to explode because it has a cell wall, but it's going to be full and tall. It doesn't tell us anything about what happened here. And then it tells us, well, those in the 5% plasmolyze, which, if we recall from a previous thing, right, that means that that water moved out, causing, which makes sense because it's going to go from a high water concentration. This one had a 98% water concentration inside, right, 100 minus 2, and 95% water solution outside. So we're going to, water moved out, causing it to shrivel and pull away from the cell wall. All right, now drawings themselves. So even if you have this, that's not going to earn you points on an FRQ. You have to turn these pictures back into words. So that's where it's important that we know our super sassy science words and can use them correctly. Explain the movement of water between the cytoplasms and the surrounding solutions. So the only ones that it talks about are the 0 and the 5. So for this first one, you could say, in the 0% solution, water moved from the hypotonic solution in the beaker to the hypertonic solution inside the cell, causing the cell to swell and become turgid. Because it said explain the movement of water, right? So explain is like how or why. Don't just tell, like, why did it move, right? Because there was the hypotonic to hypertonic to try and get to equilibrium. Similarly, or inversely, in the 5% solution, water moved from the hypotonic inside of the cell to the hypertonic outside of the cell in an attempt to become more isotonic, resulting in the cell membrane shrinking, pulling away from the cell wall, and plasmolyzing. All right, so, right, that's when we add our super sassy science words. So you gotta practice writing. I've got resources for FRQs for this. You can check out in the pinned comment below. Then B, predict what would happen if the plant cells were instead placed in an isotonic solution. Well, guess what? They were in the middle. It didn't tell us what happened. It's asking us to predict what will happen. So if you have an equal concentration of salt on the inside and the outside of the cell, water is going to move equally in and out. There will be or you could say there's no net movement of water. So it doesn't mean water is not moving at all. It is, but for every one that goes out, one will come in. All right. And so, and the cell will stay the same size. So predict what will happen. What will happen is water will move equally into and out of the cell, and the size will not change. So those are the main things to consider. Practice, practice, practice. Practice writing, practice drawing, practice all these things until you get comfortable, because you're gonna see a cell in a beaker question on the AP Bio exam. If you found this video helpful, don't forget to give this video a like, subscribe to my channel, and leave a comment, a question, or an emoji. Teachers, check out Teachers Pay Teachers. My store there is Know and Grow with Gnomes. I have tons of resources for water potential. I'll bundle everything, FRQs. So head on over there. Keep learning, keep growing, and we'll see you next time.